Mount Zion Metric Higher Secondary School, Department of Biology. This is a 12th Biobotany practical session. So I am happy to welcome you all to this practical session. So I hope you will enjoy this practical video. So there are 3 slides in this session. We are going to discuss the slide number 1, TS of mature anther. Then slide number 2, LS of angiospermic ovule, and slide number 3, TS of Nerium leaf. These 3 things, 3 slide preparation technique that we are going to learn now. At first, the TS of mature anther we will discuss. So first one, this is a Dardura metal, Oomathampu, and this is a flower, trumpet shaped flower, and yellow colored stigma is there, then white colored style, and here a white is back like structure present. This is called anther. Okay, these are called anther, and when we cut this anther in transverse section, we will be getting a butterfly shaped structure. It is called a TS of anther. Okay, so this is a vase glass, this is a slide and cover slip, brushes, then slide, permanent slide is ready, and glycerin, and this is a microscope. So in the exercise 1, TS of mature anther, aim to study and identify the given slide TS of anther. Principle, androsium is made up of stamens. Each stamen possesses an anther and filaments. Anther bear pollen grains which represent the male gametophyte. Requirements: anther of Dardura metal, glycerin, safranin, slide, cover slip, blade, brush, needle to prepare temporary slides, permanent slides of TS of mature anther, and compound microscope. So these are the things which are required for the preparation of temporary slide. Diagnostic features of TS of mature anther. A mature anther is bilobed. Bilobed means it is having 2 lobes. One lobe is this, one side, right side, and left side having another lobe. Okay, so how many lobes are there? A mature anther having 2 lobes, right side and left side, one lobe. There are 2 lobes are there. So therefore it is called dithecus. And 2 lobes are joined by a connective tissues. These 2 lobes are joined by a connective tissues. Each anther lobe has 2 pollen chambers. So each anther lobe, this is one anther lobe, and it is having 2 pollen chamber. Here one will be there, and here another one pollen chamber is there. Then here one more, and here one more. So totally 4 pollen chamber is there. So how many anther lobes are there? 2 anther lobes. So pollen chamber it is having 4. Each anther lobe having 2 pollen chambers. So in which the pollen grains are produced. So pollen chamber la வந்து pollen grains வந்து produce ஆகும். A microsporangium or pollen sac is surrounded by 4 wall layers. They are epidermis, endothelium, middle layer, tapetum. So epidermis is the outermost layer. Then endothelium is the second layer. Okay. Then third layer, brown color இருக்கக்கூடியது endothelium. Yellow color இருக்கக்கூடியது middle layer. Then blue color இருக்கக்கூடியது tapetum. Okay, tapetum layer. These are all the 4 layers of the microsporangium. A center of microsporangium, a pollen sac is filled with the haploid pollen grains. So in the center of the microsporangium, so it is filled with the haploid pollen grains are present. This is the regarding the diagnostic feature of TS of mature anther. So in the exercise 2, LS of angiospermic ovule, aim to study and identify the LS of angiospermic ovule. Principle: the in female reproductive part of a flower, the basal swollen part is ovary. The ovules are present inside the ovary. Later they develop into seeds. A requirement: permanent slides of LS of ovule and microscope. So this is a pistil, a female reproductive part. The first part is said to be stigma, then style. This swollen part is called ovary. So inside the swollen part of ovary, ovules are placed. Here there are 6 ovules are placed here. After fertilization, these 6 ovules are converted into 6 seeds. Okay. So if you take a longitudinal section of this ovule, we will be getting the structure like this. Okay. So diagnostic features: ovules or megasporangium is protected by 1 or 2 covering called integuments. This is a ovule. This is a structure of ovule. It is protected by 1 or 2 outer covering. This ovule is having 2 outer covering. This is the inner integuments and this is the outer integuments. The stalk of the ovule is called funicle. This is a stalk because the ovules are attached to the placenta by this stalk only. Okay. The point of attachment of the ovule is called hilum. This region, this is called hilum. The body of the ovule is made up of central mass of parenchymatous tissues called nucellus tissues. And these are the nucellus tissues. They are the parenchymatous cells. The integuments form a pore called micropyle. This integument forming a pore, this is a small pore, it is called a micropyle. The region opposite to the micropyle is called a salazal end. Salazal end. The nucellus has large oval sac-like structure. This is a large oval sac-like structure which is present near the micropyle end. It is called embryo sac. So the embryo sac having 8 nucleus. Inside the embryo sac, 8 nucleus is present. This is the structure of mature ovule. Parts of flower, and this is a microscopic observation of LS of ovule. This is a Nerium plant and a very good example for xerophytic plants. Is an the exercise 3, TS of Nerium leaf, aim to observe and understand the xerophytic adaptation found in Nerium leaves for living in dry and xeric habitat. Principle: the plants which are living in dry and xeric conditions are known as xerophytes. So dry and xeric condition means same meaning only. The plants which are present in dry condition, it is called xeric conditions. They are called xerophytes. Requirements: Nerium leaf, few pieces of carrot, pith, blade, brush, needle, compound microscope, glycerin, cover slip, wash glass, microslide, safranin solution, petri dish, etc. Exercise number 3 la diagnostic features பாருங்க, presence of multi-layered epidermis with thick cuticle is present there in the Nerium leaf. So this is a multiple epidermis with a thicker cuticle to avoid the transpiration. Okay, to avoid the transpiration, it is having multiple epidermis as well as thicker cuticle is there. Therefore, it belongs to the xerophatic plants. Then shrunken stomata are present only in the lower epidermis. This is the lower epidermis, and in the lower epidermis, a small bit or small cavity is present. Inside the cavity, the stomata are present. Okay, again here it is a kind of adaptation to control the transpiration. The stomata are present inside the small cavity, therefore this is called sunken stomata. Okay, shrunken stomata are present in the lower epidermis. So mesophyll is well differentiated into polysac and spongy parenchyma. So here mesophyll cells, that is a leaf cells, which are well differentiated into spongy and polysac parenchyma and spongy parenchyma. Polysac means parallel shaped elongated cells ஆல் ஆக்கப்பட்டிருக்கிறது polysac parenchyma. Spongy parenchyma these parenchyma cells are filled with air space. Okay, here you could see white spaces. In the white spaces la vandu air space தான். The air filled parenchyma are called spongy parenchyma. The mechanical tissues are well developed here. So mechanical tissues in the sense xylem phloems are well developed here in this TS of Nerium leaf. And this is a diagram showing shrunken stomata. A small bit is there. So inside the bit, these are the god cell, two god cell, and here having small pore. This is called shrunken stomata. Okay, shrunken stomata is present here. Then this is a closely observe here. These are the subsidiary cells. Then here it is having two god cells. A stomata having two god cells. These pores are called stomatal pore. These are called a stomatal pore. And this is a god cell. This is a structure of the god cell. Then this is a microscopic slide of a TS of Nerium leaf, and this is a here you could see a small bit. Okay, this is a lower epidermis. A small bit or cavity is present. So inside the cavity, the stomata are present. Therefore, to avoid the transpiration. Then here the mechanical tissues. These are the xylem, phloem, which are present there. This is a midrib. Leaf oda midrib தான். இந்த midribல வந்து xylem, phloem எல்லாம் வந்து இருக்கு. ஓகேவா? Special mechanical tissues also present here. And this is a lower epidermis. This is upper epidermis with multiple epidermis. Multiple epidermal layer, then thicker cuticle also present. So that's all about TS of Nerium leaf. Thank you students. Thank you one and all for watching this video.